Okay, so let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, annyeonghaseyo, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 81st seminar, and I call it on Imperial College London Day. I just got back 3 days ago from my third European trip this year, meeting many amazing people with great minds and ideas. This week is a rare week when I am at home for almost the entire week, although I will embark on a month-long trip to mostly Asia this time to attend multiple conferences which I organize, and the EBRC meeting that is actually happening in Chicago. And then also, I also invited to give a keynote talk in Asia and also to invite seminar in multiple universities. I'm so excited to be there and meet more people, and now mostly Asian or international people attending the conference, including Metabolic Engineering 15. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Guy-Bart Stan. He is a professor at Imperial College London. As you know, and I believe he doesn't need any introduction, I guess, because we all know him. He is an expert in biosystems engineering and control, and has recently published many amazing papers on cellular burdens and their mitigations genetic circuits, which is actually one of my favorite research topics. And also I use his paper for my course class because I love those papers. Importantly, he kindly invited me for a seminar last summer when I visited Imperial College for the first time, and I enjoyed great discussion on science and life. It's my absolute honor again to meet him now virtually and host him as a pioneer speaker today. I truly thank you for your time and for your contribution to the scientific community, and also caring about young people. And that is I really appreciate it most. And the virtual podium is all yours now. Thank you.
Thank you so much, Taisok. It's actually my honor and my pleasure. And I wish, before I speak, to thank you so warmly for the immense effort that you're making to promote the next generation of synthetic biologists or engineer biologists. It's a fantastic opportunity for the young rising stars to present their work, and it's our duty in this academic life to make space for them, because they are the next generation, they are the future. So I'm sorry about that. I'm immensely honored by this invitation to be part of this adventure that you've created, and I'm extremely thankful for all your effort constantly over the weeks and the months and the years now that SynBYSS has existed. It's a fantastic thing that you have built, and I hope that it will stay for a long, long time to give the opportunity to everyone, young and old, all over the world, to participate in this adventure together. So without further ado, I'm not going to take much of the time, because I'm not the one that needs to be in the spotlight. Alex Darlington from the University of Warwick is. But I would like just to say a few words to the audience and to the generations of aspiring synthetic biologists. I've tried to collect my thoughts and to provide some answers to the question that I've received during the last few years on what does it mean to be a synthetic biologist. I don't have a good answer. I can just give a few examples of what has happened to me, and in the hope that this might be useful to others as well. So I come from a very different background. I'm actually coming from an applied mathematics background. I studied systems and control engineering. Actually, my field of expertise is control theory. And when I say theory, I insist on theory. My thesis was basically theorems and proofs all the way. And then I embarked a little bit more into questions that relate to biology. So when I joined the control engineering group in Cambridge, I started to look into systems biology. But yet again, from a theoretical control perspective. I was driven by the challenges in the field, and especially the more I read about it, the more I figured that a lot of the things that control theorists were trying to deal with in technological systems individually, separately, were all together in the field of biology. And by that, I mean, if you take challenges like the design of biomolecular systems, you have challenges all the way from data standardization to context dependencies that we will hear about today, to stochastic aspects, to robust performance when you engineer living cells or biomolecular circuits and systems. And these are things that control engineers really love to look at, but typically they look at one of these questions, one of these challenges at a time. They don't try to get to solve them or to access a problem that has all of them simultaneously. So clearly, for me, synthetic biology and what is called now engineering biology was driven by, a taste and a sense of challenge. And I wanted to be part of that challenge and to offer my little contribution to solving some of the problems that we might encounter there. My main area of interest is robust performance, or how to build systems that can perform their task reliably, irrespective of modeling uncertainties, for example, or irrespective of perturbation and disturbances that act on the system. And that's the general definition that you, or the general features that you use to justify the need for feedback control. This is what control theorists do. And they've implemented feedback control solutions to many of the technological solutions that we see emerging in our day-to-day life, from self-driving cars to fly-by-wire planes to autonomous robotics. The question that we are dealing with is, if it applies to physical systems, why does it not apply to biology? And my belief is that it does. There is nothing magical about it. It's just that the substrate we are dealing with, basically molecules or biomolecular systems, is slightly different than information-based theoretical concepts that we have from electrical engineering, mechanical engineering, or chemical engineering. And so it takes a little bit of time to put the foundations of systems and control theory for biomolecular systems, or biocontrol systems, if we can call it like this. Now, what I'm saying, in other words, is the need for a multidisciplinary approach on this. And there is currently an acute need for more engineering approaches and methods in biology, more than ever. And the reason I joined the field is because I thought that this was really important. And even in the word that we use today, we call it engineering biology. And I think that if we have a stress pattern in the pronunciation of this, it should be engineering biology. So for me, the famous quote that has been used over and over again in the definition of synthetic biology in the early days is the quote by Richard Feynman: "What I cannot build, I do not understand." And I think that this epitomizes, what we do as engineers. Sometimes, you don't need to understand all the intricacies of a complex system, but if you can kind of build one that does what you want, or even better, control it to do what you want, that's good enough. That doesn't mean that you know everything inside it. I mean, when you drive a car, you don't necessarily know all the details of how the car is built. You don't even know the equation of motion of Newton that keep the car on the road. You know your actions on the system, the steering wheel, the accelerator, the brake, and you have some feedback from the visual positioning of the car on the road. And that's enough to keep the car on the road, or heck, if you are a Formula One driver, to make it go around the laps, around the track as quick as you can. And that's what I believe it takes to build or control reliable systems that exhibit robust performance in the task we assign them to. So I want just to touch on a few key points before giving the stage to Alex. And this is probably going to be a prelude to what he is going to say, hopefully. So I think that what the field needs now is three key things. Advances in the field require a combination of skills and expertise and a willingness to stay curious in various domains. And that's something that was driving me. And I'm contacted by students or by people that are curious and ask me, what do I need to study to become a synthetic biologist? And I say, start by what you like. It doesn't matter what it is. Start by what you like. Dig deep in that field. Become an expert. If it is control theory, it's good. If it is molecular biology, it's good. It doesn't matter. Become an expert. Affirm your expertise in something you like, in your subject matter of interest. And then get out of your comfort zone. Learn new things. Move and look forward. For me, it meant transitioning from systems and control theory to molecular and synthetic biology, building an experimental lab from scratch. But we are at a stage now where, there is no such a thing as a discipline. There is blurred boundaries between disciplines, and the wealth of knowledge that we have in different disciplines is what creates the next big move forward, the next big progress. So stay hungry, stay curious, and never settle. Just know that it's good to be curious about many things. In my own academic career at the beginning, when I was applying to positions, because I had a quite eclectic profile, I was touching on many fields, people were not really sure where they could put me. And that made my selection of jobs very easy, because I said, if you don't know where to put me, I don't want to be there. I want to be in many boxes. I don't want to be in one. And Imperial took the chance on that. So yeah, stay curious. The future of the field. Where is the future of the field? Well, I mean, who knows, right? I mean, there are many possible futures, but I would say that with the advance that we see in computer science, AI and machine learning, in systems and control engineering, and in the tools that we are developing at the molecular biology level and in the synthetic biology labs, the intersection of all of that will define the future of the field. And we can envision a future where, computer-aided design, computer-aided innovation, computer-aided manufacturing will play a key role in synthetic biology and engineering biology at scale and across scales. What are the big challenges in what we want to do today? And that's really a segue to what Alex is going to tell us about today. I think big challenges are in reliable prediction, analysis, design, and optimization of biomolecular systems, and in the engineering of reliable living systems. And one of the key challenges in that is what I call context. What is context? Well, for me, and that's where I've done a lot of past research, context means, shared cellular resource, for example, it defines context. What the piece of DNA that you put inside the cell to be expressed depends on basically the whole context of the cell. It depends probably more than that on what's the growth environment of the cell, or even beyond that, what are the external factors that act on this growth environment. And all of that defines context. And this is key for this little snippet of code of life, the little snippet of DNA, to execute the function that is encoded into it. It's not a one-way program where we just read what's on the DNA and get some function out of it. This function is executed by the context itself. So understanding context and taking context for prediction and design is key. Another key aspect, which is basically crucial and fundamental to biology, is evolution. And that defines context as well. This is something that we do not have in technological systems. We don't have cars that transmutate into other things like planes or boats or other things like that. But biology does that. Biology adapts. And so mutation and selection and how to capture that in the design process is a key aspect that we need to tackle if we want to answer, bioproduction at scale over certain periods of time in a big bioreactor. How does evolution and mutation plays a role into that? Can we predict? Can we design for it? Can we start from designs that are not optimal, but become optimal via evolution as time goes by? And this is by design. So I think that these are key aspects, and this is potentially key interesting challenges that we need to take into account when we look at synthetic biology now and tomorrow. Anyway, so that was my short introduction, and I think that it is now time and my real pleasure to give the stage to Alex, and who will tell us a bit more about context. And I must say that for me, it's a great honor to introduce Alex, because I believe that Alex represents and incarnates what I was saying, the need for more engineering, the need for people that come from different backgrounds with their own expertise, with their own knowledge, and open up to these new challenges that synthetic biology and engineering biology throws at us. Thank you very much.
Thank you so much. I mean, that's absolutely brilliant talk. And I absolutely agree with you 100%. And also, I love mathematics. You mentioned earlier, start with what you like. That's also my advice to many young people, including my own teenage daughter, who is preparing for college entrance exam and other thing. But that is a key point, because if you do not enjoy, what's the point, doing it, right? So I believe that's absolutely resonate with me, and then that's a brilliant point. I really appreciate it. And then also, I love mathematics, and now I have two mathematics geniuses in the same room. So super honored to have you both. But at the same time, mathematics is actually key to synthetic biology, I believe. That's why I wanted to interact with those mathematics geniuses, I call it. And then including, you guys, and also Mustafa Kamashi, I recently visited, ETH in Switzerland. And then we had the great time talking about science and life there. So that's wonderful. So our now is time to introduce the main speaker of today with a longer introduction. And Guy-Bart, introduced him a little bit, so, that can help me and help us a lot. But he mentioned very important thing, including context and mutation. And also, sometimes, cell breakdown, also cell die. But, that is also not necessarily, that's a unique point, especially evolution, but we sometimes complain cell change a lot. But at the same time, I now realize, because my cell phone completely died for 1 week now in actually another country when I'm traveling, so that make a lot of trouble, because without phone nowadays, I cannot do anything. But, at the same time, cell also break. Cell has some context information, very important to function, and then adapt. That's kind of unique point. And that is, I believe, the Alex talk today. Dr. Alex, Darlington is leading a research group at University of Warwick, UK. So I should also call today a UK Day. Alex, work lies at the interface between systems engineering and biotechnology. He received a bachelor degree in genetics and then a master's degree in systems biology at the University of Cambridge. During his master program, he used experimental and theoretical approach. That's a rare kind of, combination in the field to investigate plasmid loss in energy-stressed yeast. Alex undertook his initial PhD training at the UK's first synthetic biology doctoral training center between universities of Bristol, Oxford, and Warwick before joining the University of Warwick School of Engineering for his PhD under the supervision of Professor Declan Bates. Interestingly, I visited all three universities to give my seminar, including one where he was the host last year. His PhD work focused on designing translational control systems to manage the impact of resource limitations, very important topic in synthetic biology, which is a common topic all three of us today are interested in. He did his postdoc research also based at Warwick with a focus on applying control theory, theoretical tools to improve the performance of genetic circuits, including multivariable control, use of tools from robust control, and developing designs for biological realizable integral controllers. During his postdoc, he won an innovation fellowship to work with Ingenja Ltd, developing new tools for pathway design in yeast. He recently won a 5-year Royal Academy of Engineering Research Fellowship, I believe very competitive one, to develop new design frameworks for the efficient engineering of biological systems within cellular and industrial constraints. He is a truly rising star with great potential to become a future leader in synthetic biology. So I'm very excited to listen to his talk today. And Alex, thanks so much for your time today. And please take it away, and it's all yours now. Thank you.
Thank you very much, both of you, for that kind introductions. And thank you, Taesok, for organizing this seminar series and inviting me to present. So if I now share my slides. Yeah, so you can see my slides.
Yes.
Yeah, great. So my research from the beginning really has been motivated by this idea of cellular constraints and synthetic circuit failure. So when I started reading about synthetic biology in the beginning of my PhD program, I was fascinated by the oscillators that oscillate without an oscillatory motif, by the activation cascades that invert rather than become activated, and by bistable switches that emerge in the absence of any actual nonlinearity that should confer bistability. And all three of those areas of synthetic circuit failure can ultimately be explained by the fact that the cell's a finite biosynthetic machine, and the more we ask it to do, the more it responds back. So that's my motivation, and that's what I'm going to talk about today. So today I'm going to talk about how cellular constraints impact engineered gene circuits, and then I'm going to talk about my PhD and postdoc work, engineering resource allocation controllers. Then I want to introduce the kind of design frameworks that I've been developing over the first year of my fellowship, and talk about how we can use those design frameworks to design control mechanisms for improving the performance of biotechnological pathways. So so my work is theoretical, although I've just started recruiting my first experimental students. But today I've removed all equations and tried to replace them with cartoons. So I hope that's appreciated by your wide audience, Taesok. So when we express, sorry, I'm just looking for the pointer now. So synthetic gene circuits often fail due to the unforeseen interactions between their hosts and circuit. So if we take this, if we take a simple gene circuit that's composed of a constitutively produced green fluorescent protein and an inducible red fluorescence protein, as we, if these two genes were completely unconnected, then as we induce the RFP, we'd expect the GFP to remain constant. That's our design intention. As we add RFP, the GFP stays high. But in reality, what we see is that as we express RFP, we get this reduction in GFP. And this angle here is essentially caused by those host constraints that leads to synthetic circuit failure. And a lot of work done by Guy-Bart and others has identified that the main course of this, the main course of these interactions in E. coli at least, is the impact of ribosome production, ribosome utilization and growth. So initially we have 2 ribosomes translating 2 mRNAs, but as we induce the RFP, we add in more ribosomes, and that number of fixed ribosomes then gets allocated across all of the genes within the circuit, and we get this reduction in GFP as RFP is induced. So here we have 2 supposedly unconnected modules where the genes are competing for shared gene expression resources, and that leads to non-regulatory interactions between those genetic modules, something that I like to call intra-circuit coupling. Now, I like to think of this as a supply and demand problem. So at constant growth rate, we've got a constant pool of resources, cellular resources here, each being utilized by one of these processes. So the process 1's consuming cellular resources through K1, process 2 is consuming resources through K2, but we get this disturbance from the circuit module back to the resource pool, which is essentially because these ribosomes are being sequestered from the circuit pool to the resource pool. And that back reaction essentially means that the input to process 1 also causes an input to process 2 through the impact of this interaction D1. So if we consider that at constant growth rate, these cellular resources, often considered to be free ribosomes, are constant, then we have a constant supply. But as circuit genes are sequestering these resources, we end up with a variable demand. So what I sought to do over the course of my PhD was essentially ask, can we design some kind of control system that means that supply increases in response to demand? So can we essentially have supply match demand and therefore reduce the impact of these 2 disturbances, D1 and D2? So the question then is, how can this be achieved? So in order to control the allocation of ribosomes, we need to think of, we need three different aspects. We need to think, 1, how can we direct the supply? 2, how do we sense the demand? And 3, how do we actuate that sensing of the demand? So essentially, how do we design a control system that lets us that lets us sense that demand and increase the production of circuit-specific translational resources? So the way we can direct the supply is through the use of synthetic 16S rRNAs, which act to specify the ribosomal proteins to specific genes. So the ribosome's targeted to an mRNA through the ribosome binding site, and by expressing a synthetic rRNA, a synthetic ribosomal component, we can render a portion of the ribosome pool quasi-orthogonal. So we're still using the same R proteins, but instead of those R proteins being directed towards translating host genes, we can direct them towards translating circuit genes through the use of a cognate synthetic RBS sequence. So here we've got our host ribosomes translate the host's mRNA, but our purple orthogonal ribosomes translate the orthogonal mRNA. The next question, so now we're able to direct the supply of the ribosomes to the circuit. So then the next question is, how do we sense that demand? And during the course of my early research, I explored through using computational modeling all sorts of different mechanisms to test how could we sense this demand and how could we actuate this response. And today, instead of talking about all of that work, I just want to talk about the design of our final prototype controller. But if you want to read more into the different control systems that we tried, you can see some of our proposals in ACS Synthetic Biology, or we didn't get around to publishing all of them, so do get in touch if you're thinking about building these kinds of things, and we can have a chat again. So how do we sense the demand? Well, using this, we can use this idea of resource competition and disturbance in order to build a sensor. So if we constitutively express a controller protein mRNA that is translated by our orthogonal ribosome pool that then makes controller protein, then and then actuates that signal by inhibiting the production of sRNA, you can see, of rRNA, of the synthetic rRNA, then you can see here that we've created a negative feedback loop. Now, if this mRNA is constitutively expressed, then the mismatch between the mRNA production rate and the which should be approximately constant, and the protein production rate is our sensor. So here, as we express circuit genes, the orthogonal ribosomes are sequestered away from our controller protein. So the level of the controller protein falls, which means our inhibition of our rRNA is relieved. We get this burst of rRNA production, which means that we make more orthogonal ribosomes, and therefore we have matched our supply of translational resources to our demand for them. So as I said, we tried and I developed models of a number of different control systems, some focused on RNA, sequestration, some of rRNA sequestration, others based on a kind of like ribosome sponging effect. But the one that had the best performance is this topology, which we then went and I worked with Jose Jimenez, who's now at Imperial, and Chu Young Kim, who's now a group leader at KNU in South Korea. To experimentally build such a control system. So in the original open loop situation, we get a sharp trade-off between GFP and RFP production, but then when we introduce our prototype controller, you can see that we get this shallow, this line becomes much more shallow. So now as we make RFP, we lose less GFP. So we're able to allocate more translational resources as the demand for translational resources increases. And our prototype controller based on LAC-I successfully reduces this gene expression gradient by over 50%, enhancing modularity of these two, enhancing modularity and reducing the non-regulatory interactions between these genes. Crucially, and as our modeling predicted, we actually have quite a lot of control over the performance of this controller. So we find that we predicted that by changing the exclusively one parameter in the model, so that's RNA birth rate, we could tune the gain of this control system. And indeed, as we changed the induction rate of the rRNA experimentally, we validate that prediction so that it's a highly tunable control system. So having spent a lot of time thinking about orthogonal translation, the bit of my postdoc work that I now want to mention is essentially adding in that orthogonal transcription. So the question really, so I think a key question is, well, why would we potentially want to have these orthogonal transcription translation systems? And I really, this has been proposed a number of times and sort of implemented experimentally, but there's this idea of trying to, well, can we have a virtual machine within living cells? So is one way of reducing host circuit interactions the ability to completely separate synthetic circuit gene expression from the host? Now, we'll come on later to talk about probably why that won't work in the long term, but it's but we started playing around with these ideas. So if we can have, so the idea would be, well, we've got these orthogonal transcriptional resources, we've got these orthogonal translational resources, but because these resource pools are smaller than those of the host, then we're actually going to worsen our resource allocation problem. So then for a large part of my postdoc work, which we've published in Cell Systems, I've looked at, well, what should we have a control system wrapped around each of these resources in order to manage that resource allocation problem of these synthetic resources, and to start to build a way towards a true virtual machine. So we tested, so I built models of multiple different RNA polymerase and orthogonal RNA polymerase and orthogonal ribosome control systems. So here we've got one based on work by Howard Salas's group, looking at allocating orthogonal T7 polymerase using a protein-based control system. We tested ideas of using RNA, we tested in silico ideas of using RNA sequestration, and then we also looked at a non-controlled version. And whilst all of these look like they have good ability to allocate resources effectively at both the mRNA and the protein level, so we built models of these control systems. We parameterized them with biologically reasonable values, thinking about dissociation constants for things like for proteins like LAC-I or TET-R, and found that in all cases we get this good performance. So this 2D score is essentially a combined measure of how effective the control system is. So this looks great in that there's a large number of controllers that have good performance. But the issue with constructing anything in living cells is that you don't have precise control of the parameters. So we then went on to carry out a robustness analysis of these control systems using some tools from robust control. But today I'm just going to talk about our Monte Carlo analysis. So whilst to any engineers in the audience, you'll see an uncertainty value of 50% is much beyond anything that we'd expect in any physical system. In a living system, only knowing parameters to a precision of around 50% quite realistic. And you can see that any kind of uncertainty in these three control systems results in the emergence of oscillations. So although these control systems look like they have good performance, as soon as there's any uncertainty in that parameter choice, as soon as context dependency moderates that dissociation constant from being the nominal value you've chosen, but to be 1 1/3 or 1 1/2 times the value you've chosen, we're getting these control systems. Not only do they fail by having these quite large responses to disturbances, but they also we also see the emergence of these oscillatory behaviors. So we then systematically went through these control systems and removed key regulatory linkages and identified where that improved robustness but a loss to performance. And ultimately we came through to propose a new controller that utilizes exclusively orthogonal resources. So here we have an RNA polymerase here we have an orthogonal RNA polymerase that does all the transcription of both the circuit genes and its controller, and the control system. And we have an orthogonal ribosome that does all the translation of both the circuit and the controller. And what we find is that this orthogonal transcription translation system that relies exclusively on orthogonal resources can successfully coordinate transcription and translation. Should essentially be it's easily tunable with key performance metrics determined only by a couple of parameters, and it shows significant enhanced robustness, tolerating uncertainty without the emergence of oscillatory behavior up to even 80 or 90% uncertainty in the parameters. So that's where I ended my postdoc research, essentially with an idea that, well, we've experimentally we've designed and experimentally validated an orthogonal ribosome controller, and we've got potential designs of transcription translational control systems that essentially allow us to abstract away a lot of the cell's biology into these kind of modular gene expression machineries. And then I started thinking, okay, but there's more cellular resources than just shared competition for gene expression resources. So you can see here, this is the figure that I showed you initially, that we've got when cells are grown in glucose, we've got this sharp decrease in GFP as RFP is induced. And that's at fixed growth rates. My experimental colleagues, Chuyong Kim and Jose Jimenez, then said, okay, well, we'll play with the carbon source here and see what happens to these isocost lines. And what they see experimentally is that we've got this significant modification of the isocost lines. So even though the cells have got fixed growth rate and therefore we assume a fixed number of ribosomes, we get by tuning the growth substrate, we're tuning the isocost line. So that's the basis. That figure is fundamentally what motivates the rest of my fellowship and my ongoing research, which is, so it's we're dealing with a problem that's more than RNA polymerases, more than ribosomes, and we really need to have whole cell design frameworks that incorporate metabolism, substrate import, product toxicity, change in cell size, the impacts of growth and potential population behaviors, be that competition or competition between different strains, or the emergence of heterogeneity in large cell culture. And the thing that's more complicated for biology is that as there's more and more work done on these resource allocation, there's more investigation into, say, ribosomal and RNA polymerase allocation is being carried out in different organisms, we're seeing that this is that these trade-offs in gene expression are very different in different microbes. So over the course of my fellowship, what my group's now working on is trying to develop extended theoretical frameworks that augment our understanding of ribosome interactions with dynamic models of metabolism to capture that metabolic and gene expression interplay. We're developing collaborations with experimental colleagues to apply these new frameworks to other industrially relevant organisms such as Saccharomyces cerevisiae and Pseudomonas putida. And really, in order to support that new bioeconomy, we're trying to move beyond the ideas of we're trying to move beyond simple GFP, RFP gene circuits, try and understand how resource allocation influences industrially relevant pathways or the new smart microbial therapies. So this is where we left off, thinking about cellular resources and allocating to processes, but what we need is a new framework that essentially adds in the blue. So what is the impact? What is the impact of growth, and what's the impact of those host processes that drive the ability for cells to produce the resources that our circuits utilize? So we need a holistic framework that captures not only non-regulatory interactions between genetic modules, that intract coupling, but also that captures the impacts of growth burden and metabolic effects, which are those interactions between hosts and circuits, something that I like to call intercoupling. So without going into the details of these large nonlinear ordinary differential equation models, I just want to talk about the kind of three key aspects of cellular physiology that we're capturing here. So and talk a little bit about the structure of the model that motivates the work I'm going to talk about in a few minutes. So the way that we're modeling metabolism, so why do we need to model metabolism? We need to model metabolism so that we can capture the cofactor supply to key biotechnological pathways like NAD or NAD, NADH, and ATP. We need those because those second messengers within the cell govern competition between pathway production and the cell's key anabolic drivers. And we also need to expand our analyses to be essentially outside of the cell, to think about that dynamic external environment and the environmental heterogeneity that comes up during large-scale industrial fermentation. We're capturing gene expression through a coarse-grained proteome that's composed of host enzymes, pathway enzymes, and ribosomes, and using mechanistic descriptions of transcription and translation that capture their substrate dependency. And always, and our models are built around that core competition for finite cellular resources. And then we're starting to expand these understandings to think about bounded import and export reactions, about mechanistic resource biosynthesis pathways and dynamic growth, both of which create complex nonlinear feedbacks or natural feedbacks. So what we're in the process of developing is a nonlinear coupled ordinary differential equation model that describes that dynamically describes the interactions between a host and circuit, accounting for metabolism, gene expression, and growth. Now I'd like to move on to some work that I've been doing using a prototype of one of these models to think about how do we design control in metabolic pathways, and that work I've been doing with Ahmed Manan at Imperial. So why do we need to think about cellular constraints on metabolic engineering? So if we just think of a core pathway model, we might think that there is a substrate, it's converted via an enzyme E* into some kind of product, and that substrate also determines growth. Now, if we look at the dependency between product formation on enzyme concentration in this small model of a pathway, we see that as we increase pathway expression, which is the x-axis, we get an increase in production, an increase in production rate. And these colors represent different kcat values. So what essentially, as this is an increase in kcat as we go from blue to yellow. And we get a linear dependency of growth rate upon production rate. But if we augment this simple pathway model with a prototype model of cellular growth, thinking about that supplies metabolic resources, that supplies ribosomal resources, and where we actually account for enzyme production values, where we account for resource consumption by both the host enzymes in black and the pathway enzymes in blue, then what we see is this quite comfortable these comfortable linear relationships begin to break down. So as we increase enzyme expression, we now get a fall in production as the cells become overloaded, and this fall gets pulled further to the right, as we lower kcat values. And not only do we also see a less sharp fall in gene. We see a less sharp fall as we increase productivity on growth, but we see that this is turning. So we get a decrease for a time, and then it turns, and we get poor growth. So we've got essentially now a regime where the cells are unburdened and a regime where they're burdened. And this kind of performance isn't captured by our simple pathway-only model. Now, one thing that this model does predict is that our cellular constraints may actually enhance our metabolic engineering efforts. So whilst we see that production of an enzyme adds additional burden, so here in the black dotted line, we have our protein burden, and then the colored lines represent different kcat values. So for if that protein was actually an enzyme, you can see here that as we increase kcat and we sequester resources and we sequester metabolic resources to a pathway instead of host growth, we get an added burden that's additive to the protein expression burden. What we actually see when we look at the cells cell's internal dynamics, or the model cell's internal dynamics, is that substrate consumption is shifting our cells into an enzyme-dominated proteome. So on the x-axis here, we're increasing enzyme production rate, and the dotted line represents the case where it's a protein, not an enzyme, and the solid line represents the case where it's an enzyme. So we're actually getting that flux distribution away from central metabolism. And what we can see here is that as we produce enzyme, we actually get higher levels of enzyme at lower induction rates, because those cells have been shifted from a ribosome dominated proteome. The stress of our pathway shifts them into an enzyme dominated proteome that acts to enhance enzyme production. You can see here that as we shift there, we actually get a concurrent increase in the host cells and in the non-ribosomal cellular proteins. So that's the other host proteins, but also the cell's enzymes. Those two things together mean that as we activate our metabolic pathway, our in silico cells respond by increasing our substrate production rates, enhancing our pathway performance. So here, in that situation, it's actually looking like, for once, host circuit interactions might be aiding our process. So constitutive production of metabolic pathways is often what's utilized by industry, but there's increasing interest in developing control systems that switch cells from a growth state into a production state. And this is the work that I've been doing with Ahmed Manan, who's at Imperial. And what we do here is, in the growth phase, we invest in cell number, so the pathway is inactive and the cells are just growing. And then after a period of time, the pathway is induced such that the pathway is induced, so now we shut off all of those other cellular processes that are utilizing cellular resources, and rather we just have this essentially shortcut from substrate import to protein production, to product production. So we've been investigating this with a host circuit interaction framework. So I just want to start off by thinking about that basic idea of a dynamic control system versus a static design system. Now, the rest of the work that I'm going to present is based on these two key industrially relevant metrics of yield, which is determining essentially how much of your product, how much of your substrate in is at your output, which determines your economic cost, viability, and volumetric productivity, which is looking at how much have you produced over the total production time, and that's another key determinant of economic productivity. So depending on your process, depending on your pathway of interest, you might need high productivity and accept lower yield, or vice versa. And then we've been using multi-objective optimization to design pathways and control systems to optimize for both of these objectives. So if we think of your traditional metabolic engineering approach, which says, how do I tune my host enzymes and how do I tune my pathway enzymes? How do I tune my host enzymes and how do I tune my pathway enzymes? Then within our in silico framework, we get this gray trade-off curve. So as we increase yield, we get reduced productivity, and that's a standard result to see in these situations. And then we've augmented that pathway with a dynamic control system, which actively switches the cells from that growth state to that production state. And we see a significant increase with a maximum productivity more than doubling. And we really shift this curve up and to the right. So we're showing here that that these dynamic control systems have significant improvements to both yield and volumetric productivity, and that's the base case here that I want to compare our next few slides to. So what does this control system look like? So the blue line is topology 1. And the way that our control system works is when the system's induced, we inhibit the host's enzymes and we activate the pathway enzymes. So we're essentially switching the cells from making ATP or energy and switching them into making exclusively protein. Now, if we then considered, well, what about these two alternative topologies where one regulatory link is removed? So in topology 2, our control system seeks to only activate the pathway upon induction, and in control system 3, we inhibit the host enzyme upon induction. And what we see here is that these two other topologies, topology 2 and what we see here is that topology 3, where we only control that native branch rather than this two branch point, rather than both sides of the branch point, sorry, has nearly the same performance as topology 1. So topology 1's our standard approach, activate the pathway, inhibit the host, whereas topology 3 seems to give us the same level of performance, even though it's only based on the idea of inhibiting the host. And this is a consequence, and we've seen this because of a consequence of our host circuit interaction model. Oh. So if we then dig into the dynamics of our simulations, looking at when we have the same volumetric productivity, we can see that as the and then we look at the actual dynamics of those processes. So here the solid lines, the blue lines represent that initial topology 1, and the yellow lines represent that topology 2. Topology 3, which has similar performance. And our solid lines are the host's enzymes, the dotted lines are the pathway genes, and then this light gray lines, or the light blue, light yellow lines, are the upstream pathway genes that are unregulated. So what we see when we look at their transcription is that shifting our cells into the energy stress state by stopping production of by reducing production of that energy supply, increases expression of expression of both the upstream genes and the pathway genes. So as we reduce the amount of energy in silico cells are producing, because we're shutting off that energy production growth pathway, we're shifting the cells into an energy stress state, which means that we're enhancing production of both the pathway genes and the upstream genes that are supplying substrate to our pathway, and that's all through non-regulatory. And in the blue context, that's through a regulated interaction here, but in the yellow context, that's happening purely because we're shutting off pathway genes. So that's a consequence of these shutting off host pathway genes. So that's a consequence of these host circuit interactions. And what we see here, if we look at the number of proteins per cell, is that our synthetic protein E star ends up at a similar concentration with the two, ends up at a similar concentration of the two control schemes, even though there's no direct regulation in the case of of E star in the case of the yellow traces. And as we look at the on the right hand side, bottom right hand side here, if we look at the flux after the induction, whilst there's a different set of intermediate dynamics in the long term, this topology 2 is achieving similar increases in flux to topology 1, which albeit at this more elongated response time, and that acquisition of the same flux is giving us that same volumetric productivity, even without all of those additional regulation steps. So we then extended our analysis to think about the other genes within the model. So we thought, well, if dynamic regulation, if non-regulatory interactions are leading to this upregulation of these upstream enzymes leading to increased substrate production, then can regulating those upstream enzymes actually improve performance? And so we added in that dynamic regulation here, dynamic regulation. So this black line is our original system, and then these three colored, and then the yellow line is where we activate the upstream genes. The blue line is where we inhibit the upstream genes upon pathway induction. And the purple line is just constitutive metabolic engineering to overexpress those upstream genes. And what we see here is that the additional regulation is unnecessary. So yes, to get from our nominal case of our black design, which is topology 1, to the an improvement in volumetric productivity could be achieved by engineering the constitutive expression of those upstream genes, but actually regulating those upstream genes is unnecessary. They can just be constitutively overexpressed. And then we also thought that we also turned our attention to product export. So most synthetic products lack a decent transporter to get out of the cell. So one way of enabling cellular export is to express a synthetic transporter that's more suitable to the product you've made. Now, obviously, this expression of a synthetic exporter creates an additional burden on those cells. So we then sought to say, well, should we only produce our exporter when our pathway is active? So that's our dynamic synthetic exporter T star. And what we show here is that whilst dynamic regulation of T star of that export protein does improve performance, actually the cells are quite the cells tolerate quite a significant exporter. So for example, this one is 4 times the size, where adding the dynamics gives us no improvement, or gives us negligible improvement when you consider the genetic engineering efforts in order to achieve it. So we think really what we can conclude here is that a constitutively expressed exporter is not going to is not going to be overly burdensome, unless of course that exporter is 20 times the size of a normal E. coli gene. So really what Ahmed and I have shown is that if we have a control system regulating a transcription factor that has the opportunity to regulate upstream enzymes, host enzymes, pathway enzymes, or pathway export mechanism, you don't need to regulate with activation or inhibition all of these steps. You just need this one in blue. So we're essentially saying, instead of editing 4 or more genes here, we can probably get a lot of performance out of just focusing our experimental engineering efforts on that key node that's taking substrate and diverting it to growth, rather than all these other nodes in the network. So in conclusion, we're developing my group's developing host pathway frameworks to enable systems level design. We're developing these methods for key industrially relevant organisms and focusing on adding in additional industrial constraints. So we're thinking about outside of the cell as well as inside it. And we're exploring how dynamic control strategies can be used to improve performance of gene circuits and metabolic pathways. And all of this work is and then by working with experimental groups, we're using these methods to enhance biomanufacturing as part of a greening of the chemicals industry. So I'd like to thank you very much for listening. I'm now 1 year into my fellowship. I'm still trying to grow my group. So if anybody has any mathematical, computational, or experimental students who are looking for PhD opportunities, please do urge them to get in touch. And thank you very much.
Oh, absolutely great talk. And I'm also glad I see many people I know with your collaborator I'm talking about. And Ahmed will talk about his part, I believe, later this year. So I'm excited to listen to his talk. So, Guy, do you have any comment or question to him?
I have a couple of questions, yeah. So the first one, I think that you might have addressed, but I want to make sure I've understood this more clarification on my side than a question to you. So when you were looking at the orthogonal pool of ribosomes, you were talking initially about the work where you consider the orthogonal 16S subunit. But then it's not really orthogonal, is it? Because
no,
then
so because you're still relying on all of those R proteins. So yeah, so it's not a truly orthogonal ribosome in the way that you can have a truly orthogonal polymerase. But what it does give you is that ability to subdivide the cell's ribosome pool. So that system is never going to reduce burden, because burden is about total ribosome sequestration. But what it will do is reduce host circuit interactions by enabling you to dynamically choose dynamically match the circuit's demand with translational resource supply.
That is because we have actually what we have by design here is on the proviso that the growth rate is fixed and therefore the total ribosome pool is fixed. We have split a constant ribosome pool into two subsets.
Yeah.
Where one of them uses the native and the other one the orthogonal 16S subunit. So we have basically a second conservation law in this subset, but which means that overall, if the size of the subsets of pools is smaller, you might actually hit a harder constraint.
You do, and you see experimentally that the orthogonal ribosome. You see experimentally that the orthogonal, like if both genes are using the orthogonal ribosome pool, the isocost line gets steeper.
Yeah.
If you divide your gene circuit between, so one is host and one is orthogonal, you can manipulate the isocost lines that way. But yes, so you make your resource problem harder, but you gain control over it.
Yeah, unless you completely overwhelm the whole ribosome pool.
Unless you completely overwhelm the whole ribosome pool, and then the cells will die anyway. Yes, of course.
Yeah, cool. And then the other question that I had was in the model that the work with Hamad. And you were doing the modeling. One thing that I wasn't completely sure I followed. So in your model, you have added basically a layer of complexity that allows to predict the impact on the growth rate, essentially, and also predict the growth rate from the different things that you take into account at the level of the shared cellular resources.
Yeah,
but I wasn't sure. Is the growth rate then impacting back on the dilution rate?
So the growth. Sorry, carry on.
One of the key problems that we have in this model is that there is this circularity where the dilution impacts the availability or the size of the resource pools, but at the same time, those resource pools usage impact the growth rate. So it becomes a bit difficult to juggle with these things normally.
Yeah, so what we have there is we've got a mechanistic description of growth. We've got a phenomenological description of growth as a consequence of total protein synthesis, which is changing all of your growth, which changes all of the cell's dilution rates. Which then feeds back on your substrate production, which feeds back on your mRNA birth rates, including the birth rates of the ribosomal genes. So we've got that mechanistic feedback loop. We've got that feedback loop there, mechanistically captured by the equations without without a phenomenological description. So it's hard to point to the equations and say there is the feedback. But it's there.
It's embedded.
Yeah.
And when you look at this, do you take into account biosynthesis of resources like ribosome biosynthesis or other things like this via the pathway, for example?
So we've got at the moment within that prototype framework of microbial growth, we've got resource autocatalysis, so the fact that ribosomes are synthesized and then make more themselves. So yes, the resource catalysis.
I was asking because we just released a paper on this
about 3 weeks ago. Yeah, I just saw it.
With Kirill, right? With Kirill Seshkar. And I mean, it's not exactly the same, but it has similarities. And I think this is very, very interesting. This is typically very difficult to take into account But when we put that in place, we realized that a few of the laws experimentally observed, well, laws, I don't know what to call them. It's basically the observation, and then from them they extract what they call laws, were conflicting. And if you take these things into account properly, they are not conflicting, actually. They are a result of the interaction that you have between growth rate and consumption of shared cellular resources. And they become explained stricto sensu by that. So yeah, it's quite interesting, and I'm more than happy to discuss with you. I'm very interested in what you have done there.
Yeah, no, I think that's a great conversation to have another day. What I really like about the paper that you've just put on bioRxiv is that you've managed to reduce what's a very large model down to those nice descriptive equations. So there's obviously a tour de force of algebra behind there by your student, but I think it's nice to actually get some. It's nice to have a single growth rate equation out of those kind of out of those ribosomal.
I think we come from the same school of thought. I mean, I think there are two main. I mean, there are more than, but in my mind, there are really two main school of thoughts in modeling: the minimalistic model school of thought, and let's model everything we can, like the big models that you see that try to model every possible reaction in the cell. Being a control engineer, I like you, I really like minimal models. So for me, a model, I mean, the main quote for me is that all models are wrong, but George Box. And this is really important because I use models to control cells or to control processes. And my use of model is not mechanistic. I don't need to explain everything that is happening. I just need to explain what I can observe. And the simplest possible model that does that for me is useful in the sense that I can then do, what we do in this paper, which is derive analytically solutions or do optimization or those. I'm just not good enough to do it with big models. That's the reality. I just don't do that.
I think the challenge is that when we take those smaller scale models and we then talk to, say, an industrial partner who says, oh, can I use your model? This is my pathway. It's got 5 different shared cellular second messengers, and I need you to also account for extracellular growth rate, extracellular substrate coming into it, and you just end up with, and you start sketching it out, and then you end up with a huge thing that you can only study by like death by simulation approaches, and then the problem is the numbers matter. So I think we are at this challenge of complex enough to capture the biology to be useful industrially as part of that synthetic to engineering biology transition. But those models are not trustworthy because the parameters aren't known, where we've got the small scale models that are small scale models give us, like you say, you can get an analytical insight there. I suppose the question is, I think it's still unknown what level of model do we need to effectively engineer, say, a complex metabolic pathway?
And
engineer the dynamics of that complex metabolic pathway.
But a lot of, it's totally true, and the rigorous answer lies into model reduction technique that would allow you to cross between the different granularity layers. But this is hard. The thing that I would say is that when we build physical systems, there is a bit of both. But to get to build things that work, at least at my level of comprehension of complex systems, I much rather have, a kind of pseudo-phenomenological, or depending on how you use that, pseudo-mechanistic model, whatever you want to call it. Basically a simple minimalistic model that allows me to understand when I turn this dial, something will happen in the right direction, than a big model where I have thousands of parameters and I have no clue which one would give me what I want. So the problem, of course, is the boiled down minimal model. The parameters they have are also probably not necessarily easy to relate to the actual kinetic reaction rates or whatever true parameters of the physical or the biological system. So that's really the conundrum in which we are. But when we started building cars and planes and other complex systems, we started with minimal models initially. Now, in parallel with that, we built big simulators that are ultra realistic and where we can test all of the things. But I think these are kind of two parallel things. They are not basically meant to be the one and unique, all-encompassing modeling techniques.
I think you're right. I think sometimes
the two, right?
And I think the thing, it's interesting talking about at the beginning you stressed engineering in your definition of engineering biology, and I think sometimes because we have such a group of collaborators as a field, we have a group of collaborators that's such a diverse background. I think sometimes there's a tendency for some to say, well, what is the model? And the argument that we need to make successfully is that, well, we build cars and aeroplanes with many models, all of different complexity. And there can be no one model. Yeah.
There is
I agree with you.
Actually, I think it's not from me, but I was at a dinner sometimes at some point, and this question came up with, can you give me the model or the best model? And the answer was, no. What do you want to do? Tell me what you want to do, and I will build you a model for it.
Yeah.
And that's really the point. If you need to do something, then we can talk about granularity of modeling. But if we don't know what we do, then there is no such a thing as the model.
Yeah.
A model for design, control, and optimization is not a model for explanation or for hypothesis testing. They are very, very different. And I'm not even going to open the Pandora box of inferring causality via modeling. So I mean, that's one thing that I wanted to cover. Another thing that I thought was really cool and that you presented in your talk is looking at the whole process, at the whole bioreactor layer, basically taking into account the growth environment and taking into account then what's happening when we need to model this whole thing, not just the cell that we engineer, but the whole process, consider what the impact for bioproduction is. And there, I'm not making that on purpose. It's just serendipity has it. We also some time ago on bioRxiv, and soon it's going to come out now, looked at this interplay that exists from a modeling point of view of how to capture evolution, how to capture mutation, when the driver for it is burden.
Yeah.
In the sense, when you engineer your cell with a new piece of DNA or a new expression system that has little benefit to them, it impacts their growth, like you have shown in your modeling techniques, and then if you take that into account and overlay on it, basically a model, a very simplistic model for evolution, which is, there is a probability or a propensity to mutate any piece of DNA that you insert in the cell, or basically any part of the DNA in the cell. Then basically you have now a model for a cell that takes into account resource, that takes into account growth rate, that takes into account external parameters like substrate availability and so on and so forth. But then it's not one, because this cell basically then makes a copy of itself.
Yeah.
Duplicates itself, and each time this happens, there is a propensity for mutating.
Mutation, yeah.
Then you have a mutation, and then the model that you use can be used to infer the growth rate of these two cells, which might be the same as the original cell or might be different, we don't know.
Yeah.
If they have mutated, the growth rates are different. And as soon as you have cells with two different growth rates that use the same substrate to grow,
they're competing for substrate.
That's right. And then the one that grows faster takes over.
Yeah.
Because when it makes another copy of itself, the progeny again will grow faster. at the same growth as the mother cell, and so they will take over. So there's a principle of mutual exclusion, and that basically is a route towards modeling heterogeneity in bioreactors and to take into account basically what happens when you have a diverse population of cells that comes up, and it boils down. We show that it can boil down simply to taking into account burden.
Yeah, in the design space.
Instead of, different dynamic behavior that you wouldn't have otherwise, which is quite interesting. I mean, so yeah, I think this is basically looping back on my introduction and the whole talk that you have given on the importance of really understanding context and understanding what biology does every day in a way that we are not so familiar from an engineering point of view.
Sure. So any other comment, question?
No, that was it. This is the main thing that I mean, I really enjoy your talk. I mean, of course I'm biased because it's a little bit my
sure.
Yeah.
Yeah, so I now see, my time 11, almost 30. I have 2 more questions from audience, so I want to answer this question, and then I actually have the question, but I will, wait for the informal, chat without recording. So Maya asked 2 questions, and the question is, what numbers are you using for orthogonal systems kinetics? Are orthogonal systems efficient enough that you can build this immediately, or do this depend on future advances on, I think what is ORIDO and OR, I believe the polymerase, right?
So thank you for the question. So my main part of that project was the controller design. What my experimental colleagues found is that the orthogonal ribosomes that they had in their hands were weaker for protein expression than the host RBS, and there was also I'm trying to remember now. I think I'm trying to work out, qualify my words correctly. We had an amount of crosstalk that was not too great to prevent the project going ahead. So the orthogonal ribosomes we have are orthogonal, but they are weaker, and there is a level of crosstalk, as in the host ribosomes still translate those mRNAs, those orthogonal mRNAs. But we included that in the modeling of the supplementary material, in the supplementary of the paper, and it doesn't that doesn't affect things like controller design as long as you account for it. But yeah, so in the we accounted for them being we can account for them being weaker, and we can account for that level of crosstalk, which I think answers your first question.
So the wonderful. So the second question is, in this very, she said, not real question, interesting one, however. Has anyone tried circularly permuting and fusing R proteins for fun to achieve a ribosome with fewer distinct proteins? Her initial impression is ribosomes are too complicated, unknown in both biogenesis and translation dynamics to do it in any rational way.
So I know what people have done experimentally is staple. Well, they've said what they call it, like a molecular staple, where they tie the large and small subunits together. So you render the small subunit orthogonal using the 16S RNA, and then you can express another RNA whose sedimentation coefficient escapes my brain, to staple that large and small subunit together. So that would I suppose that would render your orthogonal ribosome entirely orthogonal, except for the fact that you're still relying on the host to synthesize them. I don't think anybody has been able to make essentially a lightweight, small-scale ribosome that can only that can function on its own. So the beauty of the bacteria of RNA polymerases is that bacteriophage have spent millennia evolving small discrete polymerases that we can then bolt into synthetic circuits. And I don't, whether that would be possible for the ribosome or not, is probably an open question, but it is a very large complex. So
yeah, that's very
I would
interesting.
guess not.
Yeah. So I guess now I see 1.5 hours basically. So I probably going to close, and then we will chat if we want to more, basically without recording. So let's close. So thank all for joining and staying today. We'll meet again on May 25, Thursday, the same time, the same Zoom link. We'll have Professor Michelle O'Malley at University of California, Santa Barbara, and Dr. Neil Darby at Harvard Medical School, so back to US from the UK. As usual, the follow-up informal chat will occur without recording. Please stay here. If you are interested in chatting with today's speakers, I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stopped recording. Just give me 1 second.